Okay. Okay. So this topic is quite complicated and quite controversial, and beyond that, it is also very, very important. As we nowadays we know that it is the fastest and growing neurodevelopmental disorder along with the maximum prevalence, though it may not be the fastest, but yes, prevalence is increasing very, very high. So do we need to investigate each and every child which comes to our clinic with the diagnosis of autism? So how do we need to titrate and how we need to tailor down the investigations? So we'll just try to, at the end of this webinar, we'll try to figure it out, how we can investigate our approach, narrow down depending upon the presentation and other things on the spectrum child when it presents to us. So we'll begin our talk with a very important statement. We know that autism is a neurodiverse condition. So no two children are ever same on spectrum. So if we make them stand, 100 kids are standing in a row, no two children will be ever same on the spectrum. So, and this is what exactly, it is a very famous statement which was given by Dr. Stephen Shaw, that if you have met one person with autism, you have met one person with autism. That means the presentation is totally different. Each and every child cannot be, tailored to a same approach. It needs to be tailored down as per the needs and as per the findings or as per their abilities. So as a result of that, there are no standard guidelines as of now for investigating an ASD child. So investigations need to be tailored down as per the etiology, the clinical profile, presentation, and associated comorbid conditions. So it's very, very important that clinicians need to spend an ample time building rapport with the parents, counseling them and convincing them regarding the rationalization of doing the test as advised by the doctor or the clinician. Because if we can, there are ample battery of tests and we want to write the test, it can be a complete list of tests with their whole prescription. So that is why it is very, very important to counsel them, rationalize them, and build them a rapport with them that why you are doing these investigations and what is the rationale behind them. Because they will be consuming a lot of energy, they will be consuming a lot of money, financial, everything is there. And also the mental state of the parents is also not good. So these things need to be taken into consideration when we are planning an investigational approach for a child. Now, before going into the investigations, we should know what is the etiology of ASD. So now a lot of debates and everything has been going around, and we know that after so much research and everything, nowadays we do talk about a lot of things, what is known as epigenetics. So broadly, the etiology of ASD has been broadly classified into three types: environment, epigenetics, genetics, and the fourth most important thing which is now coming is nutrigenetics and nutrigenomics. The way how our food express the representation of the genes and how our genes are influenced by the diet what we take. So both of them is vice versa. So along with this, now we are talking about nutrigenomics and nutrigenetics. We can't do much about genetics, but we can do a lot of things about environment, partly about epigenetics. So taken into consideration about all this broad etiology of ASD, the ASD symptoms fall under this category of repetitive behavior XYZ, the core symptoms. So these are now being classified as the broad etiological classification of ASD: environmental, which may include maternal diet, which starts right from the conception before the child is born. Though unfortunately, the presentation of ASD comes generally at the period of 2 to 3 years, with the earliest presentation of socialization skills or delay socialization. Rarely you might get regression, but generally it is a socialization, generally it is the language delay or the speech and language delay. So environmental factors, epigenetics, genetics, and then nutrigenomics and nutrigenomics. So these are some of the etiologies for the ASD. Proposed, I won't say it is the etiology, these are some of the proposed etiologies of ASD. So if I come about the first, the environmental factors. The environmental factors which starts right from, I'm just touching on these because I'm not going to the details as we have done a separate webinar on this. So environmental factors, the environmental factors which affect the gut microbiota of the mother. That is, these are some of the factors which are present antenatally. So how these environmental factors can influence the gut of the microbiota of the mother, which can indirectly affect the gut microbiota of the child. So the environmental factors antenatally or conceptually are the maternal diet. That is what we exactly talking about, nutrigenomics and nutrigenetics. So what about the maternal diet? What is the mother taking balanced diet? Is it rich in iron? Is it rich in calcium? Alcohol intake, smoking history, exposure to radiations? All these things need to be taken into consideration when we are working on the environmental factors. The use of drugs, valporate, XYZ drugs, which may affect and can be teratogenic. The second most important, another important thing is along with the maternal diet is infections, bad obstetric history. How about the history? Was the mother on antenatal? Was the mother on antibiotics? Was there any bad obstetric history? So, or preeclampsia history. So all those things need to be taken into consideration when we are working on the investigational approach. Then mode of delivery. A lot of research has been done and it has been proven beyond doubt that the mode of delivery also increases the probability of going for a child into an ASD phenotype, not genotype, phenotype. That is what we talk about epigenetics. We'll be discussing on that. So a child who is born with a normal vaginal delivery will carry all the beneficial microbiotas from the mother's vagina into the gut of the fetus of the child. That is predominantly consisting of lactobacilli and bifidobacteria. Whereas those which are born through the cesarean section, they'll be carrying most of the microbiota which is present on the skin of the mother. That is strepto and staph. So it has been found that those who have been born through the vaginal delivery, the chances or the probability of going into ASD is less. Said that, it doesn't mean those children who are born with normal delivery will not go into ASD, and those who are born with a cesarean will go into ASD. It's only increases the probability of going to this. Then type of feeding, breastfeeding versus complementary feeding. The first and foremost important thing is in the breastfeeding, we have a undigested carbohydrate which acts as a prebiotic is HMO, human milk oligosaccharides, which plays a very, very important role acting as a prebiotic in influencing the growth of selectively bifidobacteria. They have specific receptors. Again, I won't go into the detail. They have specific receptors which specifically increases the growth of bifidobacteria compared to any other type of prebiotics like FOS and GOS, which are synthetically or naturally available plant sources or inulin. They increases the growth of both bad and good. So this is a great, mode of feeding is very, very important. That is why exclusive breastfeeding for the first 6 months or beyond that, along with complementary feeding, also plays a very, very important role as an environmental factor influencing the gut microbiota of the child. Because when we go on a formula feeding, they won't contain HMO, they'll be containing FOS and GOS as a prebiotic. So these are some of the things, and as a result of all these factors combined together, they can cause alteration in the gut microbiota. And this alteration in the gut microbiota, which is known as gut dysbiosis, can express or can express the phenotypic presentations of the gene expression. That is what we call as epigenetics. I'm using the word phenotypic expression. Why I'm using it? Because it doesn't affect the genotypic expression. The DNA sequence is same, but the phenotypic expression of the person is changed. That is what we call about epigenetics. The major difference between epigenetics and genetics is in genetics, there is a change in the DNA sequence. Whereas in epigenetics, if we go for the exon sequencing or WAS, that there won't be any changes because there is no change in the DNA sequence. When we are going for whole exome sequencing or etc. and etc., they detect the DNA sequence changes. So they will pick up the genetic changes, but they won't be able to pick up the epigenetics. So this is what about the environmental factors. Now, as per this, there is a proposed model of ASD. What this is, because of these changes which are presenting in the antenatal in the mother, affecting the gut microbiota of the mother, XYZ reasons which we have declared, the diet, the feed, the infections, the use of antibiotics, the bad obstetric history, all these things causes the activation of immune system of the mother known as maternal immune activation. Among this activation of maternal immune system, there are different interleukins are there, interleukin 1, 6, 7, but the latest which has been done, and even this study has been done by Harvard University, and they have found that interleukin 17 is one of the most important cytokines or the interleukins which has been found to be raised in mothers with all these presenting features, aggregating factors causing maternal immune activation. When this maternal immune activation happens, it causes the alteration of the gut microbiota of the mother. And because of this alteration of the gut microbiota of the mother, the same gets transferred to the fetus, causing the gut dysbiosis, and that gut dysbiosis in the fetus can cause or influence the epigenetic presentation of the child or the gene, thereby resulting in ASD. So this is some of the proposed model of ASD on the basis of nutrigenomics, gut dysbiosis, and epigenetics. So factors which can affect the MIS, maternal immune activation, could be antibiotics, advanced maternal age, prenatal infection, stress, pregnancy, and complications. So going little beyond that, I will be talking about the GBA, the gut-brain axis. What exactly, how this epigenetics and causes the influence in gut microbiota and causes the brain changes in the ASD kids. So gut-brain axis, it has been proved beyond doubt that yes, gut-brain axis is one of the most important factors which may be involved in majority of the neurodevelopmental disorders, whether it may be schizophrenia, whether it may be Parkinsonism, whether it may be Alzheimer, whether it may be autism. So what exactly happens? Epigenetics, the XYZ factors, they causes the gut dysbiosis. When there is a gut dysbiosis, which may be caused due to XYZ reasons, which we have discussed, that is the type of feeding, the mode of feeding, or the stress, the environmental factors. So when there is an alteration of the gut microbiota, that means to say the balance between the good and the bad gets disturbed. When that disturbs, it causes an excess release of toxic metabolites known as SCFAs. Now, SCFAs are short chain fatty acids. If we talk about SCFAs, they are actually very, very good. They help in reducing the redox potential. They help in mitochondrial inflammation, mitochondrial improvement. They play an important role in mucosal barrier amendment. They help in preparing an important role in maintaining the T junctions between the intestinal cells, help in mucosal absorption, maintaining the mucosal integrity. Then why they play an important role, why they show a negative effect. Now, among these, the most important are propionic acid, butyric acid, and glutamate. Now, among these, propionic acid is the major culprit. See, we know that anything good and anything which is there, which is in excess can be harmful and in less can be harmful. So it has to be maintained in an optimum level. So there is an optimum level of all these SCFAs like propionic, butyric, etc. And all these are produced by the lactic acid producing bacteria like lactobacillus, etc. That means they are beneficial. So SCFAs are good. But the problem is the propionic acid. It is produced in excess amounts by the Clostridia. And Clostridia are the non-beneficial or the bad bacteria. So what they found in children with gut dysbiosis, when they did the studies for SCFAs, I will show you the investigational charts, how they have been shown. The levels of propionic acid were very, very high. And when they correlated with the microbial diversity of the ASD kids taking the stool samples, they found the Clostridia levels of the non-beneficial bacteria were high. So as a result of this, when the propionic levels are high, they causes release of all these inflammatory cytokine, that is the cytokinin cascade gets activated, especially interleukin 6, all these XYZ, T nuclear cells, TNF factor. And when they causes an activation of these, there is causes an affection in the HP axis, and there is an increased release of systemic circulation of these inflammatory markers, which causes the blood-brain barrier, and they affect the what we call the microglial cells of the brain. The microglial cells of the brain act as the macrophages. They help in synaptic pruning. Now, when the macrophages, synaptic pruning means the unwanted synapses needs to be cut down, which generally happens by the age of 2 to 4 years. And that is the role of microglia. That is the microglia which are known as the natural macrophages of our brain. So because of this cascade and all these gut dysbiosis, the microglial activity gets reduced. Because of that reduced or defective microglial activity, the synaptic pruning is affected. And all this because of the synaptic pruning is affected, the neuroinflammation and the core symptoms of ASD comes. So this is the crux, and in a very simplified way, how this GBA presents and how this epigenetics, gut dysbiosis, and brain, that is the synaptic pruning, is affected. So in a small summarization of this slide, gut dysbiosis plus defective synaptic pruning could be a possible of ASD. It's not that it causes ASD. Nobody can comment on that. But yes, this is what our understanding is nowadays coming. MIA, maternal immune activation, alteration in the gut microbiota, the type of delivery, LACS of VSD, NBD, causes the alteration in the gut microbiota in the child, which can cause the cascade, defective macrophage functioning, causes defective synaptic pruning, and the appearance of an ASD phenotypically in the child. So now we have talked about the environmental factors. So let's talk about what is epigenetics. So in recent years, epigenetics modification has considered to play a major role in mechanism of autism spectrum disorders. Epigenetics is the study of phenotypic changes caused by dynamic and reversible changes in gene expression, while keeping the underlying DNA sequence unchanged. So that is the major difference between an epigenetics and genetics. It's very, very simple. It's just imagine that we have a car is there and there is an exhaust pipe, and some notorious child comes and puts up the rod in the exhaust pipe. The car will not start. You try ample number of times, the car will not start because the exhaust pipe is blocked. So that is, and when you remove that rod from the exhaust pipe, the car starts. So that means it is an epigenetic factor. Phenotypically, the car was not getting started because we have introduced some change in that, but there was no malfunctioning. Genetic change was not there. But if there is a damage to the exhaust pipe, no matter you put the rod or anything, the car won't start. So that is what is the difference between epigenetics and genetics. So DNA sequence remains unchanged, but over a period of time, if these epigenetics changes continues to take place, it may go into an irreversible stage. And that is probably a reason to explain that why ASD kids over a period of time may improve or some may not improve. If it would have been purely genetics, no matter whatever interventions you do, whatever interventions, therapies or medications you give, they won't have happened any improvement at all. It is probably because of this reason that children over a period of time may show some improvement depending upon their abilities. In adolescence, they might be better compared to now, maybe in the future 3 years, by the continuous use of therapies, because these are considered to be an epigenetic changes more rather than that of pure genetic changes. In humans, the main mechanism includes, so what are the main epigenetic changes which are there? DNA methylation, post-transcriptional histone modification, and miRNA modification. So the three most important epigenetic factors which has now been considered in ASD model is DNA methylation, histone modification, and miRNA modification. So let's discuss a bit about that. As an environmental factor, gut microbiota can trigger the epigenetic modification in the host, thereby because of the reasons of XYZ, which can affect and bring about the epigenetic changes and derived in the metabolites. So that is what I was telling you. Nutrigenomics or nutrigenetics, gut dysbiosis, epigenetics, ASD. So these are the three most important: DNA methylation, histone modification, miRNA modification. So what is DNA methylation? DNA methylation in a very simple word is adding a methyl group to a DNA molecule, and this is what is called as DNA methylation. Now this addition of methyl group to the DNA molecule is very, very important in order to carry out hundreds and 200s of enzymatic actions in our body. So over the past 3 decades, there has been sustained research activity devoted to correlate the methylation patterns in with ASD as a result of advancement in next generation sequencing technologies. So what happens exactly in DNA methylation? Now, normally what happens, the methyl group which is there, the methyl group gets bound to the DNA nucleotide known as cysteine, which is a general binding of the DNA. This is a normal binding of the methyl group on the DNA molecule cysteine. But sometimes, instead of this methyl group binding to the cystosine, it may bind to some other nucleotides. And because of that, there is a phenotypic expression or the defective DNA methylation, thereby expressing the phenotypic changes in the child depending upon the variety. Now, why the child gets present into different categories of ASD, might be mild, moderate, severe. Now, that totally depends upon how severe or abnormal the binding of methyl group to the DNA structure. Sometimes the cystone is the normal. It may bind just close to the cysteine to the other molecule. The presentation might be not that severe. It may bind little more beyond to the cysteine, another nucleotide, which may be far away from the normal cysteine. So as a result, there the presentation phenotypically might be little more severe. That is what the explanation as per the epigenetics molecule of DNA methylation. There are so many other thoughts of ABA, behavioral, sensory, and etc. But this is one of the school of thoughts in related to epigenetics. So high levels of non-CpG methylation in the brain plays a very critical role for neuronal development, and that exactly highlights in the spectrum in ASD kids. So several environmental factors, including in utero exposure to tobacco, folic acid, primary vitamins play an important role in methylation. And 80 to 90% of children diagnosed with autism have methylation impairments, irrespective of the fact whether they are bound to little more severe abnormality of the structure or less, or maybe close to the cytosine. But this is very clear that in 80 to 90% of the children who are in the ASD, the binding of methyl group is not happening at the cytosine. So a growing body of research indicates that methylation influences the development of autism. So there are variations in MTHR gene and the presence of folate receptor autoantibodies. We might have heard about something what is known as cerebral folate deficiency or CFD. Nowadays, if you see, it has been found to be an association, and a lot of literature and papers have been written on cerebral folate deficiency in relation with autism. So what is this MTFHR gene? So MTFHR gene plays a very important role in the production of methyl tetrahydrofolate reductase. Now, MTFHR is a gene. It is a rate limiting step which plays an important role in the conversion of 5,10-methyl tetrahydrofolate reductase to 5-methyl tetrahydrofolate reductase. Now, folate. Now, this is the active form of folate. Now, what we take through the diet is folic acid and etc. with the or maybe in the form of supplements. But folate is the one which is the active form, and the active form of folate is 5-MTHF. So this deficiency of this MTFHR can affect this pathway, which plays another important role in this methylation pathway or DNA methylation. So the frequency, that is why whenever we are going for the epigenetics test nowadays, we need to look into the activity of MTFHR, what is known as MTFHR polymorphism. Now, this gene will only express when it is homozygosally expressed. So there are two variants like C6770 variant. Another variant is there, I think C663 variant. But the important variant is C6770. It has to be present homozygotically in order to present this disease or in order to express phenotypically. If it is present in a heterozygous presentation, MTFHR or this pathway will not be affected, as it is a recessive gene. So then you have the folate receptor antibodies, which also plays an important role in methylation. And what happens exactly in this cerebral folate deficiency? There are folate receptor antibodies are there which prevent the binding of this folate and going into the brain. As a result of this, what they have found, the children with folate receptor antibodies, the folate is not present in the brain. So they need to be provided with an active form of folate which can, cross or bypass these blocked receptors, and that is what is called as the folinic acid. So these are some of the studies and some of the literatures where they have found that in children where there is a cerebral folate deficiency, giving folic acid will not be useful because it is not the active form of folate. We need to give an active form of folic acid, that is folinic acid, and that too it should cross the blood-brain barrier bypassing this folate receptor antibodies. So if I go little in the summarizing, the environmental factors which can affect the gene expression, that means epigenetics, or if I take about the DNA methylation, they can be in utero exposure to medications, drugs, prenatal influences, multiple gestations, bad obstetric history, infections, perinatal factors, fetal growth retardations, and intrapartum hypoxia. So all these factors can affect our expression of genotype. Epigenetic expressions can be affected in a way DNA methylation can be affected. So this is the bad trio of MIA, dysbiosis, and ROS. So environmental factors in the mother in utero, XYZ reasons, they causes the MIA, maternal immune activation, which in turn causes the activation of the cascade pathway, which causes the gut dysbiosis, and that gut dysbiosis in the mother causes the epigenetics changes in the mother gene expression, that is DNA methylation, and ROS, that is known as reactive oxygen species, that is free radical production goes up and all those redox potential values goes down. So this typically gets transformed into the fetus, and then it can present into later life as a presentation of phenotypic ASD. So if I go about a little more deep into the two cycles, this is a very complicated cycle. Even it will be very difficult for me to explain, but just I'll take some bits of it. This is very, very important to understand when we talk about epigenetics. So folate cycle and methylation cycle, or the DNA methylation, which we have talked, is a very important pathway in epigenetics. So when I talk about this methylation cycle, there is a cycle in our body which keeps on rotating as a part of methylation cycle, where MB12s or methyl groups are being released. So if I go in this diagram, there is MSB12, that is MB12 or methyl groups which are continuously being released in this cycle. These methyl groups get bound to a particular structure of this, as I have told you, cytosine, which causes a DNA methylation part. So as was I telling you, that is DNA methylation. So this MB12 along with this MTFHR, which is methyl tetrahydrofolate receptor, along with this B12, this MTFHR activity cannot take place without the production of this MB12. And this MB12 during the binding of this DNA that is at the level of cytosine is very, very important for the activity of MTFHR to take place. Now this methyl tetrahydrofolate reductase plays an important role in the conversion of 5,10-methyl tetrahydrofolate reductase to 5-methyl tetrahydrofolate reductase. Now this F5-methyl THF is an active form of folate, and this should cross the blood-brain barrier and reach the brain in order to get adequate levels of folate in the brain. So when there are folate receptor antibodies which are present in the blood-brain barrier, as shown in this diagram, this F5-methyl THF cannot cross it. So as a result, there is a deficiency of folate, which is an active form. Actually, it is 5-methyl THF deficiency in the brain. So it is known as cerebral folate deficiency. Now, many a times we might go for the blood levels of folic acid. That will not give you a true levels because we are not considering with the peripheral blood levels, we are considered with the brain levels. So in studies, they have shown that we can do a spinal LP, but that is generally not been advised and not been done also as per the recommendation. So these are the two most important cycles which we need to understand when we talk about epigenetics. And as per the literature, 50 to 70% of the kids on ASD might have folate receptor antibodies. How that clinical presentation? It's very, very difficult, but yes, aggression, irritability, anxiety, without any reasons, continuous disturbances, aggression. We might be doing a lot of therapies. We might be doing XYZ things, but in spite of that, if there is continuous regression, if there is continuous improvement is not happening, so all those things need to be taken into consideration when there is an oxidative stress in the body. So to check that, the most important text is ROS, that is called redox potential. That is the ratio of reduced glutathione to oxidized glutathione. Now, among all these, when the crux of this cycle is the end product of this cycle of methylation cycle is glutathione. Now, glutathione is the natural antioxidant in our body, which plays a very important role in mitochondrial activation, mitochondrial functioning, and all the important enzymes which are acting as coenzyme like B12, B6, B9 in the cellular pathways or in the enzymatic pathways. So the end product, the glutathione, is not being produced in an effective levels when there is a defect in the DNA or methylation pathway. As a result of this, the levels or the free radicals levels in the body is very, very high. So there is a reduced oxidized levels, and there is high oxidized level and reduced glutathione levels in the body. So as a result, the ratio of reduced to oxidized goes low. Another epigenetic mechanism what they talk about is histone modification. Now, histone modification, we know that histone is a protein which is present in the chromatin, which plays a very important role in shaping and functioning of the chromosomes. So they have found that this modification of histone can also affect the shaping and the presentation of the chromosomes, though genetically it may not be affected. Suppose a chromosome is like this, it might go like this, it might go like this. So as a result, the functioning is affected, but genotypic presentation remains the same. So, but as of now, it's just purely on the research. It's purely on the literature. There is no clinically we can do anything to identify these histone modifications. Then miRNA modification. This is the third epigenetic. miRNA are the small 18 to 14 non-coding RNA molecules which transcribe in a tissue-specific manner to regulate the target genes. Now we know that RNA is a highly unstable structure. As a result of this, it is impossible to detect the RNA levels in the blood. And it is because of this reason that many of times, majority of the vaccines they are DNA based, and only rarely one vaccine, Moderna vaccine, which was RNA based. So probably the reason is it is a highly fragile molecule or a structure. So, but now what they found, this miRNA specifically plays a very important role in cellular processes, including cell survival, apoptosis, aging, differentiation, carcinogenesis, and metagenesis. And this also plays a very important role in post-transcriptionally regulating the gene expression. So what they found that in last decade, various studies have determined the role of miRNA in etiology of ASD, and several studies indicated that there is an altered expression of miRNA in different autistic individuals. The normal presentation of miRNA is disturbed in variety of autistic individuals in their studies and literature which they did. So as a result of this, what they found that one of the studies which was conducted by Hung et al. Found that there was a differentially expressed miRNA presentation was prevalent in high percentage of male autistic patients. So now miRNA can be detected in saliva, serum, plasma, and blood as they are relatively stable. So this is the unique peculiarity of this miRNA, that it is relatively stable. As a result, it can be a useful diagnostic tool or a useful biomarker for an early detection of ASD. So recent studies indicated the potential use. It's only a potential use, not recommended as of now. So potential use of using an miRNA as a biomarker for ASD. So if I take about the complete crux of all this, you have a diet, nutrigenomics or nutrigenetics, which can affect the expression through the XYZ environmental factors, causes an alteration in the gut microbiota, causes the gut dysbiosis, which can affect the epigenetics of the gene expression, that is epigenetics, through three most important mechanisms: DNA methylation, histone modification, histone acceleration. So these are the epigenetic modifications which are happening, ultimately landing the child into an ASD presentation. So this is the example of epigenetically regulated genes has been found and detected in various studies which has been done associated with ASD. Among them, the most important is MECP, which has been found to be associated with Rett syndrome. And these are the alternations in the miRNA presentation. So these are the different phenotypic expressions of the miRNA in various ASD kids, which they found on the various studies and databases and MIT net database, which they did. So this is the studies which has been now being done, doing, and regular research is being doing on epigenetics and how is its relation with epigenetics with ASD. So the role of epigenetics in autism spectrum disorder has been supported by evidence from various different research studies, but it's still a quite complex cycle. And it's, I would say it's too early to tell that, yes, we can have some definitive tests to go for that. So, but yes, the epigenetic mechanisms are quite mysterious and quite complex, but yes, they may definitely open some future avenues in order to understand the pathologies and genetic and epigenetic expressions, environmental factors, and how they're related to ASD. Now, I have talked about environment, I have talked about epigenetics. Let's talk about genetics. Now, if I talk about genetics, 49.8%, total is 62% has been associated with genetics. But frankly, when we go for the genetic analysis in an ASD child, I believe out of 10, maybe hardly 2 or maybe hardly 1, we will be able to detect some genetic association. And majority of them, a whole exome sequencing or CMA, chromosome microanalysis, comes out to be normal unless and until if there is an associated genetic history in a sibling or XYZ. So if I talk about genetic association, 62% has been associated. And out of this, 49.8% are considered to be inherited common variants. That means which has been transmitted from the parents. Now, 2.6 considered, 2.6% are considered to be heritability, inherited variants. 9.5% they consider to be de novo variants. That means which are not being present previously in the parents and not being transmitted. So this is the overall presentation of how the genetics can express in a child as an association of a genetic factor for ASD. So environment 38.1%, which we have discussed in our previous slides. So let's go a little bit more about the genetics. So this is the clinical profile which can present in an autism spectrum child. They can be metabolic, they can be motor issues, they can be seizures, they can be sensory issues, they can be GI gut disturbances, they can be immune impairment. We know that they are very much prone to the allergies, they are more prone for adenoids, they are more prone for frequent infections. And even the studies have found that children who are in ASD, they are taking 2 to 3 more times the course of antibiotics compared to the non-ASD kids over a period of a year. And this study was done by GSK in order to promote the use of, their probiotic in order to use along with the probiotics. Along to use with the ASD. So this was a very good study which was done by GSK. So what are the indications or guidelines for genetic testing? Now these guidelines, there are different guidelines as per the ACAP, as per the ARP. So I'll be sharing all the guidelines in relation to the genetic testing, because many a times we get a question, do we need to go for a complete genetic analysis or complete genetic testing for every child on ASD who comes up to our clinic? So that probably these slides will answer our question for a child who is coming to our clinic with a diagnosis of ASD, that to whom we go for a genetic testing, to whom we do not, because it is quite expensive. Cost is also there, finances is involved. So we need to look thoroughly whether this candidate is the right person to go for genetic testing or not. So there are different organizations, and as per them, they have different recommendations. So if I go for as per the recommendation of Autism and Intellectual Disability Committee of American Academy of Child and Adolescent Psychiatry. So these are the guidelines which has been taken from various research. I will share you the references also. So as per the recommendation, they say that CMA in all individuals with global developmental delay, intellectual disability, and or with ASD. So as per them, all children with ASD, with or without ID or GDD, should go for genetic testing. FMR1, that means we know that fragile. Repeat analysis in males and females with ID or a family history of ID. So depending upon the history and physical examination, we do tend narrow down our test P10 if the head circumference is more than 2.5 standard deviation. MECP testing for Rett in females with severe ID. So they have clearly mentioned MECP testing should be done for Rett syndrome in females with severe ID. Karyotype if chromosomal syndrome is suspected. So this is as per the guidelines of ACAP. So if I go for the guidelines for American Academy of Pediatrics, they say if a comprehensive history and exam are indicative of a specific syndrome or disorder, proceed with specific testing in ASD, with ASD and or without ID or GDD. So they clearly say that without specific history, without any indicative findings, you need not go for a genetic testing for every ASD kid. So if all individuals with ASD and IDD without specific forming, consider CMA and FMR CGG repeat analysis. So these are the guidelines as per the different organizations. Then if I go for the guidelines for International Standard Cytogenetic Array, ICSA, along with the American College of Medical Genetics, what they say that CMA, chromosomal microanalysis, is the first genetic test in patients with GDD, ID, ASD, or multiple congenital anomalies. So according to this forum, they say that all ASD kids should go for CMA, that is the first genetic test which they should consider. And then you need to narrow down as per the detailed history and etc. So you can take the snapshots. I won't be able to read each and everything as the time is not there, but these are the guidelines as per the different organizations. So as per the American College of, I have told you CMA is the first genetic which should be done. Then multidisciplinary committee on panel on this genetic, they say that should be done in all individuals with ID or ASD. If ES is not diagnostic, then does not include copy number, ES variants, then accordingly you need to go for the CMA if is needed. So as per them, they are the different guidelines which they have suggested, and they have categorized the categories to which genetic test the one should go. So what is the purpose of going for this genetic testing in patients with ASD? First of all, the most important thing, whenever you are planning for this genetic investigations, you need to approach clinically also in that matter. First and foremost, you should have a complete pediatric genetic evaluation, a person who is very well, familiar with the genetics. So you need to sit down with the genetist, and then you have to need to discuss all those things. So comprehensive history is very, very important. 3 generation family pedigree chart should be involved whenever you are testing for genetics or planning for genetic testing in individuals with ASD. Then you have physical examinations, considering are there any growth parameters, defects, are there any anthropometry defects, head circumference, micro or macro, or skin examinations, which may be neurofibromatosis, tuberous sclerosis, as they are common genetic associations. So we need to look into those genetics factors and clinical picture of neurocutaneous markers. Then accordingly, deciding on these first two steps, then you can proceed for the laboratory test, whether you go for CMA, whether you go for FMTR, whether you go for P10, whether you go for WES, and then accordingly we need to taper down. So this is as per the genetic testing. So if I take the crux of all this, a genetic testing is only indicated if there is a family history positive, if there is an associated clinical finding consisting of syndromic features or some metabolic features or some neurocutaneous markers, then only genetic testing should be done. It is not recommended to be done in every child of ASD coming to our clinic. Now, when we talk about the metabolic testing, now metabolic testing, as this is again as per the American Academy of Pediatric guidelines, the prevalence of mitochondrial disease in ASD is estimated to be about 5%. That is approximately 500 times more than found in the general population. So the prevalence of abnormal metabolic biomarkers is even higher, suggesting that as many as 30% of children with ASD might experience metabolic abnormalities. So when we are considering for the metabolic abnormalities, what are the initial tests we need to consider? Serum ammonia, lactate to pyruvate ratio, and the levels of many other mitochondrial markers like specifically carnitine and pyruvate, which are basically gives you an indicators for mitochondrial dysfunction or mitochondrial malfunction. Remember, it is not a mitochondrial disease. It's just a mitochondrial dysfunction. So this is the literature, and this as per the references which they have suggested along with the American Academy of Pediatric guidelines. So ARP guidelines for metabolic testing says the yield of routine metabolic testing for children with ASD is low. ARP stands for American Academy of Pediatrics, low and not recommended for regular use. So as per them, they say that it is not recommended on regular use. So what are the indications that we suggest that yes, a child on ASD should go for metabolic testing? However, they also say, however, large population-based studies are lacking, so accurate prevalence and diagnostic yield estimates are not available. So they don't confer that it should be not done, neither they say it should be done routinely. So what are the investigation, what are the clinical criteria, or what are the history suggestive of where metabolic testing or mitochondrial testing should be proceeded? So these are some of the workup when we should go. If there is a history, this is the directly I have taken from the ARP guidelines, so there is no change in the word. History of atypical regression when there is later than 2 years of age, motor regression or multiple regressions. Family history of early childhood death or diagnosed with metabolic disorders, specific like tyrosinemia, cystonemia, and XYZ, or maybe galactosemia and XYZ reason. So phenylketonuria, which is also very important. So physical features such as significant hypotonia or weakness, like visual or hearing impairment, dysmorphic features. Children who present with motor delay should be evaluated with creatinine kinase and thyroid stimulating hormone. So these are some of the specific things which they have mentioned. And many a times I feel that children who are on ASD, they do have a lot of core issues, core strengthening issues. So definitely, invariably, motor delay will be present in majority of the kids with ASD. So so what are the tests which we need to do? These are the initial battery of tests. Fasting blood plasma amino acid levels, urine for organic acid levels, acyl L-carnitine metabolite levels, and testing for specific metabolic disorders like tyrosinemia and phenylketonuria if there is an associated positive family history related to that. Now, as of now, there is no evidence at this time for routine testing of hair, blood, or urine for environmental toxins or heavy metals outside of laboratory screening for lead exposure. So as of now, the American Academy of Pediatrics recommends only for lead testing, screening for lead testing, whenever a child comes for an ASD. Other than that, no other heavy metals like cadmium, mercury are recommended. Heavy metal testing is only and only recommended if you are suspecting a case of from an high risk exposure, that is near the factories, or a factory or industrial employee who is working in those areas where they are exposed to some, all these heavy metals. So this is the recommendation which has been given by American Academy of Pediatrics. Now I come on to the gut dysbiosis. So I am taking the etiology step by step, and accordingly I am trying to explain what are the things investigational approach we can do as per the presentation, because the presentation might be so varied in every child, it becomes difficult. So as per 2017 American Academy of Pediatric guideline, they say gluten and casein IgE level testing is beneficial in subset of ASD kids showing symptoms of intolerance. So routine testing is not recommended. So this is very, very important, that routine testing of routine testing of this IgE levels of gluten and casein is not recommended. It is only recommended in some particular subset of ASD kids where the parents complain or show symptoms of intolerance. Like they can be excessive bloating, they can be excessive constipation if the mother is, the child is taking too much milk or casein. So all those things need to be taken, and if the mother gives, okay, so I stops the milk and it becomes little better, there is continuous bloating, constipation issues, in spite of resolving by giving laxopegs or peg leg and etc. So those things, then you need to consider that yes, it might be an intolerance issue in that child or insensitivity. So now how this is related to this, as I've discussed, the autism, the altered gut microbiota, the toxic metabolite, the leaky gut, the neuroinflammation. So this all is a vicious cycle. This figure I have taken from British medical journal, from British Medical Journal, BMJ. It's a very good article, and it was a review article, and as well as it was on a meta-analysis which was done. And on the basis of that, what they found, what are the early biomarkers of leaky gut in ASD? So there are some of the biomarkers which can detect that yes, a child might be having possibility of leaky gut issues. Leaky gut issue is what? Leaky gut is a very fancy term. It basically means there is a problem. There are T junctions which are there, TJ junctions which help in binding of the mucosa. So when these TJ junctions get separated, or they get breakdown, or they are not joined, the mucosal integrity gets disturbed. So leaky gut compromises of effective TJ junctions, decrease in the mucosal integrity, decrease in the absorption of mucosa, and decrease in the levels of crypt heights, or what we call as crypt hypoplasia. So there is a decrease in the surface levels of the crypts. Now exactly what one particular term is there, I'm not remembering. So that exactly leaky gut means. It compromises of all these biochemical changes which take place at the level of molecular changes which take place at the level of the mucosa. So in order to detect these leaky gut changes, what are the biomarkers which can help us? These are calprotectin, zonulin, occludin, CD14 precursor. Now, for your knowledge, in India, there is no testing which has been done for all these except for calprotectin and zonulin. Only these two levels have been done by some particular laboratories in India, like Lalpath is the one which is being doing. So calprotectin levels and zonulin levels are being done by Lalpath. And other than this, I don't think any other labs is doing this test. But occludin and these are some of the other markers which has been done, but in India, I don't think they have been doing. So calprotectin levels will be increased, whereas zonulin levels will be decreased in a child suspected with a leaky gut, which can be a very good biomarker. Zonulin is more sensitive compared to calprotectin because calprotectin is it is more specific marker for the purpose of inflammatory bowel disease. So it can be raised in Crohn's, it can be raised in any other inflammatory conditions. But when I talk about zonulin, it is more sensitive and specific towards the leaky gut in ASD. But calprotectin can also give us an invariable biomarker testing. So SCFAs, I was telling you. So SCFAs, short chain fatty acids, what these are quite beneficial. They help in increase water absorption, maintaining the increasing motility, help in increasing the availability of mucosa to the cells, improving the tight junction, the TJ junctions, and improving the villi height to crypt depth ratio. That is what I was exactly trying to explain you. This ratio gets disturbed when there is gut dysbiosis, when there is leaky gut. So when there is a gut dysbiosis or leaky gut, the SCFAs, the ratio gets disturbed. As I was telling you, too less is bad, too high is bad. So in this SCFA, the major culprit is propionic acid. Whenever there is a gut dysbiosis, the propionic acid level shoots up. And invariably, if the propionic acid level shoots up, it indirectly states that the clostridium levels in the body are high, in the gut is high. Why? Because propionic acid is specifically produced by Clostridium very high amounts. It is not being produced by any other bacteria in excess amounts other than Clostridium. Now, this is a stool comprehensive analysis in one of ASD kids. Now, this unique test, the stool comprehensive analysis, what is the peculiarity of this test? The first of all is that it gives you a complete description of all the different fatty acids, short chain fatty acid like acetate, butyrate, and total fatty acids and propionate. Now, this is the reference range. For example, if I go for propionate, 11 to 25 is the normal range. If this level goes beyond 25, it is a direct indicator for me. Yes, there is an excess levels of propionic acid in this child presenting with gut issues to me. And yes, it could be probably an indicator for leaky gut in my child. So gut dysbiosis is there. I need to work on the gut dysbiosis. How we need to work, that is a different topic. So we are just right now working on the investigation part. So again, we can look for the acetate, butyrate, and then a unique thing is total SCFAs. Now, when we go for the total SCFAs, the majority of them is being calculated on the basis of acetate, propionate, and butyrate, because valerate is in very, very less amount it is present. So the total SCFAs is also very, very important in correlation with the propionate. Okay, so if the total SCFAs goes beyond the reference range, along with a high propionic acid, this indicates that the gut dysbiosis is disturbed. And another thing is that they will also give you a balance between how what is the percentage of good bacteria, what is the percentage of bad bacteria. As mentioned, commensals, beneficial, and dysbiotic. Dysbiotic means the non-beneficial like Clostridia or yeast and etc. Then clinically, you can detect on the basis of Bristol stool scale, which is a very, very important scale, which has been we do use, especially it is being used by GI persons. So clinically, you can detect that what type of stool is there. Type 1, type 2, and type 1 and 2, they are considered to be constipated stools. Whether it is separate hard stools, whether it is coming as a sausage shape, then what is the type of variancy. So on your clinical history, you can mention as per the Bristol stool sample or as per the Bristol stool scale. As per the history, it is suggestive of type 2 or type 3 or constipated or diarrhea. So this also helps in giving you an analysis about the severity of the stool or the severity of the gut disturbances. So to go for the testing, GI questionnaires, which I have told you, a very important good questionnaire which can help us to identify is that is the Rome 3 criteria, revised Rome 3 criteria for functional gastrointestinal disorders in young children and young adolescents, So I use it sometimes for my, for my patients when I used to do. So that will give you a complete idea about the clinical history as per the parents, that what is the consistency of the gut in terms of stools, in terms of associated problems like bloating, constipation, diarrhea, and etc. Because there is a scoring is there which they tell you, as per then you can mention the severity is mild, moderate, and etc. So for GI test, you go for comprehensive digestive studies, Bristol stool sample, GI questionnaires like room 3, and calprotectin along with zonulin, etc., which I have told you, plus along with stool comprehensive analysis. Then what is the role of neuroimaging? It is another big question. Do we need to do an imaging in every child presenting to us? So as per the American Academy of Pediatric guidelines, incidental findings are very common in neuroimaging studies obtained in the workup of children diagnosed with ASD, but rarely provide etiological information or require intervention. Means to say, all these imaging, definitely there might be some subtle changes, but are those changes significant enough to disturb our intervention? No, because in majority of the times, whatever intervention we are doing, they will remain the same unless and until there is some gross change. So as per the ARP guidelines, in majority of the children with ASD, there won't be any gross change in the neuroimaging. As a result, routine testing is not recommended. So imaging may provide only valuable insights in the neurobiology of ASD in relation to the thickening of the cortex, the trajectory of the brain. We know that there is what we call the synaptic pruning, the thickness of the cortex compared to the ASD and non-ASD things is varying. So that will only help in giving you a neurobiology of the ASD in research settings, but it will not helpful in clinical applications when we approach an ASD kid. So what are the indications for neuroimaging evaluation? If there is an atypical regression, if there is microcephaly, if there is macrocephaly, if there is seizures, if there is intracranial manifestations of genetic disorders, if there is abnormal neurological examination, if there are other clinical indications suggestive of needing to go for neuroimaging, maybe clot or maybe meningitis and etc. XYZ. So these are clear-cut indications for neuroimaging in an ASD kid as per American Academy of Pediatrics. So what again, EG is again a big problem. Do we need to do an EG in every child? Many a times what happens, children who are with sleep, children on ADHD, children on autism, comorbid with AHD, they might have a lot of sleep disturbances. Though they may not show frank seizures, but they do have a lot of sleep disturbance. And we go for an EG, EG will be abnormal. Now, whether that abnormal EG is an indication to start an anti-epileptic drug or not, that is the call which we need to be taken by us. Because every EG, every abnormal EG is not an indication to start an anti-epileptic drug as far as an ASD kid is considered. Because in majority of them, if there is an associated comorbidity, sleeping disturbances will be there and EG will be abnormal. But whether it is an indication to start or not, let's see that. So routine EG is not indicated in cases of ASD. What are the indications for EG? Clinical concern about seizures, atypical regression, other neurological symptoms on history examination, sudden loss of language skills with sleeping concerns. Now, this is a very, very important thing is sudden loss of language skill with sleeping concern is a very, very important marker to check for an EG to rule out Landau-Kleffner syndrome, LKS. So this is a very, very important clue which might come whenever a child presents to you at 5 to 6 years of age with sudden loss of language skills, or you might thought of mute autism with sleeping concern, that could be an indication to rule out just LKS, though it is very, very rare. So these are some of the EG indications that need to be done when to be done in an ASD kid. Now, common EG finding in ASD kid comorbid with the ADHD. Now, this is what exactly I was telling you, that if you go onto this graph, there is a graph in this that is a theta, alpha, and beta. Now, in majority of the kids with ASD, what will happen? There will be an elevated power or slow waves. Now, theta waves are slow. This is the power, the voltage. Theta waves are considered to be slow waves of 4 to 7 hertz. But the power of these slow waves will be elevated. You can see the power of these theta waves is maximum, whereas the power of other waves, that is the beta waves are considered to be high voltage, high power. But here, the power of these beta rays will come down, or what we call as the ratio, the collimated ratio of theta beta ratio comes down. So this is a typical abnormality which we'll find in an EG of an ASD kid with sleeping issues, or may not be having sleeping issues. You might go for a routine EG testing in an ASD, and you might found that the power of theta waves is elevated, whereas the power of beta waves is reduced. It's an abnormal EG, but it is not an indication to start an AAD. That is what I want to emphasize with this slide. So the potential biomarkers to differentiate people with and without ASD on the basis of differences in laboratory profiles of, these are potential biomarkers as per the American Academy of Pediatrics, but they are not currently considered to be of diagnostic laboratory test has been approved for ASD except redox potential. So platelet serotonin, plasma melatonin, urine melatonin sulfate, redox status, that is reduced glutathione to oxidized glutathione, which was considered to be a marker of DNA methylation, placental trophoblasts, and immune functions. Now Th1 and Th2. Now studies have shown that the Th1 are good cells. They help in preventing the immunity, whereas Th2, they are bad cells which help in reduce, helping in reducing the immunity. So the studies have found that there is a disbalance between the Th helper and T2 helper cells. The Th helper cells, which are good, they reduce down in children with ASD. As a result, they are more prone for immunity, immunological manifestations, frequent allergies, frequent cough, frequent course of antibiotics. So if we go for this ratio of Th1 but Th2, you will find the ratio of this comes down because of the decrease in the ratio of Th1 in ASD kids. But currently, they are no above diagnostic laboratory tests have been approved for ASD except redox potential. This is only for the purpose of potential of future test and research, but routinely they are not recommended except for redox. So to conclude, it is recommended not to routinely perform extensive medical investigation as a part of diagnostic assessment, but consider it based on clinical and physical examination. Different guidelines do not agree with regard to genetic testings. So National Institute of Health and Care Excellence, that is NICE guidelines, advises to consult with the regional genetic center and perform genetic tests if there are dysmorphic features, congenital anomalies, or evidence of intellectual abnormality. And American Academy of Pediatrics suggests testing on an ASD depending upon the scenarios, depending upon the morphology, depending upon the associated features. So consensus from the various advisory bodies suggests that genetic testing should be done on case-to-case basis rather than routine testing, as the management will not change much. So that is what I was exactly telling. Genetic testing needs to be tailored down as per the indication which I have shared you in the slides, and also as per the presentation, as per the history, as per the pedigree chart on the three generations. So investigating a child with ASD can be very challenging and exhausting for the physicians and expensive for the parents. Always spend more time in discussing the reasons for doing the test in ASD child with parents rather than spending more time in filling the prescription with tests. So broadly, we can divide investigations broadly into three levels. Level 1, which are general investigations, all routine investigations like CBC, LFT, KFT, etc., which we generally do as a baseline, though may come out to be normal in majority of the kids, except in some kids with restless leg syndrome, where there might be iron deficiencies and etc. Or there might be some breath holding spells where there might be a decreased iron levels in the body. So those are the things, but in majority of the kids, it might be normal. Other things like along with that, I have not included like vitamin D levels and etc. Though since they have a motor delay, they have a frequent history of falls. So vitamin D levels generally invariably comes out to be low in children with ASD. But said that even neurotypicals, non-ASD kids, they do also have a lot of low levels of vitamin D. So all routine investigations and as what I have told you might be normal. So level 2 investigations means specific investigations like stool for calprotectin, serum zonal levels, to check for gut dysbiosis like bloating, constipation, etc. Checking Bristol stool sample, stool for yeast or comprehensive analysis to look for yeast issues or gut dysbiosis or bad bacteria. Then blood for lead, urine, and mercury if there are frequent behaviors or low cognitive skills in spite of AB and other therapies. There is always a debate if heavy metal toxicity is a cause or consequence of ASD, though some suggest heavy metal analysis, but there are no recommendations for the same. That is what exactly I was telling you when I was showing you the guidelines for American Academy of Pediatrics. Indirect markers of mitochondrial dysfunction like L-carnitine, palmitic acid, and acyl-carnitine, which might be done depending upon case to case. So that is why we have kept in specific investigations. Food intolerance panel tests like IgG levels for gluten and casein. If there is a definitive history suggestive of insensitivity to these kids, as per the American Academy of 2017 guidelines. Vitamin and micronutrient profile, if there is a typical history of picky eating, though we know that there is a lot of issues with neophobia, texture, smelling, oromotor sensory issues. So definitely many of the kids around the stats say around 25 to 50% of the kids on ASD have picky eating behavior. So if there is a picky eating behavior and they are very notorious, particular about particular texture, we might go for vitamin micronutrient profile testing. Then torch profile in case of bad obstetric history, if there is history suggestive of that. CK levels, that is creatinine levels, if there is a cases of motor delay as per the American Academy of Pediatrics, and thyroid TSH levels. So level 3 specialized investigation, DNA microassay, genetic workup, if there is an associated history suggestive of exon sequencing, neuroimage, if there is associated history suggestive of, then you can go for exon sequencing if there is a history suggestive of, as we have discussed in the previous slides. Neuroimaging, depending upon the indications. That is why we have kept them under specialized investigations, EEG and metabolic screening, suggestive of those particular things like tyrosinemia or maybe phenylketonuria. So above are done if there is a history suggestive of genetic association in sibling dysmorphic features, or there is a history of Frank seizures. So these were the various references which we have taken. So with this, I will end my presentation for today. I hope I was able to simplify it to some extent, though it's a quite complicated and difficult topic, but still I tried at my best of my abilities to make this topic as simple as possible. Thank you, Dr. Nitin, for inviting me on this interesting topic on today's. So we are open for the questions. Dr. Nitin, over to you now. Dr. Nitin. I think Dr. Nitin is not here. Okay, I think we can take up the questions if there are any questions are there. Then otherwise we can, I think recordings will be present. And I think Dr. Nitin might be done the recording. So recording might be present. So accordingly, he can share the slides. So any questions, we can go for the questions if there are any. I think there are some questions in the chat. So name of the syndromes which are associated with the ASD. This is the syndromes which are associated with ASD. We know that these are Fragile leg syndrome, tuberous sclerosis. Then other syndromes. It can be Prader-Willi syndrome, Angelman syndromes. So these are some of the common syndromes which are associated. Then we have. So do we recommend specific diet in ASD children? What is your experience? So do you recommend specific diet to ASD kids? What is your experience? Now, again, recommending a specific diet, there is no such specific diet which we can recommend. But yes, as I've told you that if there is an intolerance to gluten and casein, then we might consider giving a GFCF diet. Then again, when I use that, yes, we might consider giving a GFCF diet. Suddenly, there might be a lot of hands raised. It is not rationalized or it is not recommended. But see, if you go to the American Academy of Pediatric guidelines, they clearly mention GFCF diet might be beneficial to some set of ASD kids where there is an intolerance. So definitely in my experience also, I have kept children on GFCF diet and I have found to be quite beneficial, provided if they have an intolerance to that. But along with that, when you are giving a GFCF diet, you need to look into consideration that you are restricting a child on the majority of the things, nutrients and so many things. So accordingly, you need to do those investigations and accordingly you to provide those supplements as per the need. It's not like that, okay, these are XYZ supplements, I give it to every child. Or this is the GFCF diet, this I need to give it to every child. Another most important thing is when you are giving a casein-free diet, you need to look into consideration. If a child is very fond of milk, how you want to alter that and provide an alternative sources. If I go into a little more detail, there are two more components of casein, A1 and A2. When we go for the normal milk, it contains both A1 and A2, that is beta-gasomorphin and A1 beta-gasomorphin. So these two are considered to be the residual or the breakdown products of casein. So when we go for an A2 milk, that is A1 free. Now A1 free is casein free because it does not produce that excess amount of A1 casomorphin, which is more harmful than beta. So when we are going for an casein free diet, you can alter for A2 milk. You can alter for camel milk, but again, those are different issues or different, topic. But yes, it has been found to be beneficial depending from case to case. Can we measure the DNA methylation level? DNA methylation level, indirectly we can measure. As I've told you, we can go for the ratio of redox to oxidized glutathione levels. But yes, in US, there are some labs are there. They are definitely doing this DNA methylation. They are doing the complete analysis of DNA methylation. That is, they are testing for epigenetics and everything. But in India, it's too early. But yes, indirect markers for us as of now, we can go for this the ratio of oxidized, the ratio of reduced to oxidized, because this is one of the most indirect and specific marker, or more reliable, not specific, reliable marker for a DNA methylation. What is the earliest stage to diagnosis ASD? What is the earliest age to diagnose dysbiosis in ASD? So there is no particular earliest age. If you talk about the age, you can diagnose autism as early as 6 months. The earliest marker to be detected in autism is 6 months. That is reciprocal imitation. When a child is sitting and you are talking to him, the child immediately laughs at you. That is what exactly reciprocal imitation. A child of 6 months who is ASD might not show that. But then earliest age to diagnose autism is 18 months, as per the American Academy of Pediatric guidelines. But you can detect early at 6 months. So similarly, when you talk about gut dysbiosis, you can detect as early as when you feel, even if a 9-month-old child is showing all those symptoms of delayed development, constipation, and you try to reduce those gut issues with reducing of milk and etc. You find the gut is there, so you can detect. So there is no specific earliest, but yes, even maybe up to an age of 6 to 7 months, you can do that. How to manage feeding issues in ASD? See, managing feeding issues in ASD is not my part. It is a part of the therapist. You need to look into the oromotor issues, that is oral placement therapy. You need to work on the oromotor sensory issues. You need to work on the neophobia. You need to work on the tongue movement. You need to work on the lip closure. So all those parts of what we call articulation issues or phonological issues, you need to work on that so that the child gets adapted to different textures and tastes. So this is more a part of an oral placement therapist rather than my part comes if there is a deficiency, if there is a history of pica, if there is iron deficiency, if there is associated problems of micronutrient deficiencies.
So there are questions on the group. Have you reached out to all? If at all there are other questions, please feel free to put it on the group.
There are some questions left, Dr. Nitin, just 1 or 2, I think, and you can let me know, then we can accordingly close the session.
Sure. Dr. Ketki has...
Yeah, it is recommended to routinely go for calprotectin tests for each and every case of ASD. Then again, probably I would say for calprotectin levels, yes, you can go, but again, you need to check as per the history and everything. But yes, since epigenetics, gut dysbiosis, and going to the brain, that is synaptic pruning, these are the main mechanisms which are involved in the etiological factors. So considering that fact, I would suggest, yes, you can go for calprotectin levels, because as per the stats, GI comorbidities are the major percentage causing the GI comorbidities are the major percentage of comorbidities in a child with ASD. Though there might be so many other OCDs, anxiety, depression, ADHD, but GI comorbidities are the major ones. So considering that fact of figure of 25 to 50%, I recommend that yes, you can go for calprotectin levels, if not in each and every child, but any child which is giving a on and off history of gut disturbances. So do you recommend psychobiotics? Yes, definitely. I recommend psychobiotics, and even I also use psychobiotics. And psychobiotics are basically different from probiotics in the sense is that these are containing specific strains of probiotics, especially lactobacilli and bifidobacteria. And even various studies have shown that specific bifidobacteria like Bifidobacteria bruvis, Bifidobacteria infantilis, Lactobacilli bacillus clausi, and Lactobacilli, all these species and have found to be very, very useful, and not only improving the gut disturbances, but also improving the sensory issues. Because through this presentation, we might be able to analyze that how this gut disturbances can affect the brain functioning, thereby affecting the core symptoms of the ASD, including sensory, including behavioral, and etc. and etc. So for this, yes, definitely, I recommend psychobiotics, and even I have found improvements in gut disturbances in children who have been long term on laxopegs, or maybe on this PEG and etc. But again, you need to tailor it as per the approach, as per the presentation. Yes, those with GI issues, definitely yes, we need to. It is advised to give this psychobiotics. Minimum age of diagnosis ASD. The age of diagnosis of ASD is 18 months, but you can detect ASD as early as 6 months. Any restriction in sugars for ASD? Any correlation? Again, this is a very controversial question. If for sugars, if I talk about, again, there are two schools of thought. Some might not accept that, some might accept. But if you go on to the ARI, that is Autism Research Institute, on this website, and go on to their research papers, they have done analysis over meta-analysis over 700 kids over a period of last 10 to 15 years. What they found, the association of sugar is not exactly sugar, it is the association of glucose. The association of glucose in relation to glycemic index, in relation to sugars, in relation to yeast. Now, yeast is a bad bacteria when we talk about gut dysbiosis. So sugar or the glucose acts as a very good medium for the yeast to grow. So as a result, when there is a increase intake of sugars or increase intake of glucose, they might be increased disturbances in the form of gut dysbiosis, which might disturb and might worsen the symptoms. But any routine restriction in sugars, I don't think it is very much recommended. But also, it is not that you can take more and more amount of sugars in excess amounts. You need to correlate with the GI issues. If there are GI issues, and if there are pictures suggestive of gut dysbiosis, definitely I would tell to restrict the intake of sugars. Then children can go for those food products which are low in glycemic index. Glycemic index, as we know, the amount of carbohydrate which we intake increases the time by which it increases sugar levels in the body. So that needs to be taken into consideration. You can go for low glycemic index, moderate, and high needs to be avoided. Earliest sign and symptoms. Earliest sign, I've told you, reciprocal imitation at the age of 6 years, at the age of 6 months. Parents do ask about the reason for autism. If no obvious reason, we see how do we counseling. See, I don't think there is no obvious reason as of now. So by the end, by this presentation, I think there are most important reasons are there, and the reasons directly probably lies in the terms of epigenetics and nutrigenomics and nutrigenetics. So if a child is diagnosed with autism, you can greatly counsel the parents for the next pregnancy that regarding the diet, regarding your happiness, regarding your emotional state of mind, regarding your intake of diet, regarding your habits while antenatally you are planning to means when the child is in the womb. So I don't think there is no obvious reason why autism has occurred. But yes, there is a very good obvious reason to counsel a parent when they're planning for the next pregnancy if their first child is autistic. So I think, Dr. Nitin, I think we have completed the questions for today. So
yes, and very extensive and very important discussion. Altogether, I think it is one of the most important and most needed discussion with a lot of doubts pouring in. I'm sure many people will have doubts and many people will be sharing their doubts with me, which will be sharing ultimately with you. So whenever ready, we will post those answers on the group itself. Thank you, Dr. Mittal, once again for taking out time from your busy schedule and keeping on the education on for all the interested fellows. Thank you, everyone, for joining.
Thank you, Dr. Nitin. And I specifically thank you for taking this initiative of this ECMS platform. Thank you so much.
Take care. Thank you.
Thank you, Dr.